Hello, this is Dr. Dan Guerra coming to you at Authentic Biochemistry Podcast Studios. We're doing a video lecture today. Today is Sunday, and it is the 9th of February, 2020. And we're going to talk about coronavirus linkage to molecular targets. So let's get started with that exactly now. All right. So, what I want to cover today is a in-depth discussion looking at the scientific literature on what are the molecular mechanisms, first of all, of coronavirus invasion. That includes transmission, that includes its replication in the cytoplasm of the host cell, and subsequent reinfection into a new host. But what we really want to do, rather than describe that in coarse detail, is get down to the molecular level, talk about what proteins are at play, how there are adapter molecules that are associated with changes in gene expression, and at what cell type are we talking about discussing relative to the invasion of the virus, because that's also very important. Most lay people, but not medical people, fail to understand that diseases can infect different cells in the body, diseases such as bacteria or viruses. And depending, some viruses can invade multiple cell lineages in the body. And the course of the events that lead to the ultimate spread of that disease is directly associated with what cells are infected and where the reproduction of the virus occurs. It's really important in terms of transmission. Also, really important in terms of immunity, whether or not the host organism, the human, that gets the disease at the individual level is able to be cured because of their immune system without the help of any drugs, any therapies other than rest and hydration. Okay? That's the typical way that you clear a virus. Your immune system clears it, and then you become immune to that virus for the rest of your life, or at least for a very long period of time, as long as there are resident memory cells that will turn on plasma cells that generate immunoglobulins. You're going to be completely resistant to that particular strain of virus. Now, bacterial infections are different. There's some similarity in crossover, Venn diagram overlap, but we're not going to talk about that today. We're really going to try to talk about the viral situation, and particularly what's going on inside cells and what kind of cells specifically we're interested in. So let's get on to that. Here's a paper that was published very recently, as you can see, Cell Death Discovery 2019 in June. So this predates this coronavirus outbreak, okay? But it's fine because what it's talking about is the SARS coronavirus. And what it's going to tell you is the SARS coronavirus open reading frame 8B. It's a specific gene that's expressed. What is it doing when it gets in the cell? It triggers an intracellular stress pathway, and it activates an inflammasomal process that utilizes a protein called NLRP3, something I talked before many times on my autoimmune lectures. Now, recall, there was a relationship of the current novel coronavirus infected pneumonia, NCIP, that's the one that's out of China right now in 2020, and the previous epidemics. The coronaviruses themselves are enveloped positive sense RNA viruses, and they are characterized by club-like spikes that project from the surface, and usually large RNA genome for viruses with a cytosolic replication strategy, as I suggested. Now, these coronaviruses cause enteritis in cows and pigs and upper respiratory tract infections in domestic fowl, such as chickens, turkeys. They can also transmit potentially generating lethal human respiratory infections from animal to human. Now, in the recent past, outbreaks of the highly pathogenic severe acute respiratory syndrome coronavirus, that's the SARS, and the Middle Eastern respiratory syndrome coronavirus, MERS, have been recognized as epidemiological agents whose close association to the NCIP make the current outbreak one to monitor. So that's why I'm looking at this paper here, because this paper has been long enough in the research mill. It's a product of the research mill that we know a lot about these other two previous coronaviruses, SARS and MERS specifically. And that's why it's good to go back and take a look at their mode of action, replication processes in general. Now, here's a paper that is published just now, January 29th, in the Journal of Medical Virology. And you can see there it's an EPUB ahead of print. So this paper will come out in the February issue of that journal. The topic of it is 2019, because it started in December, just last month or 2 months ago. The new coronavirus epidemic, evidence for the viral evolution. Now, why is evolution important? Because the NCIP is a coronavirus, which comes from the Notaviridae, which is the total order of viruses that this whole class of coronavirus comes from. The reason that it is unique is because there's been mutations, and when you get mutations and they are selected for in terms of evolution of that system, the viral system, you then have an evolutionary process that has been selected for to generate new virus that could have, for example, increased rate of transmission from animal to human or from human to human. You see, that's the kind of thing that epidemiologists are interested in. It's also the kind of thing that medical pathologists are interested in, and of course, us biochemists are, because we want to know what is the exact mechanism whereby viruses are allowed to transmit. And we need to know all the molecular patterns that happen inside the cells that are infected. So let's take a look at this paper. Now, what this paper did is used only a handful of patients, 12 to 15. So right away, it's a very small n number. Nevertheless, we want to take a look at this paper. Within the genome of the NCIP, that's the virus, after sequencing from those index patients, those 12 to 15, in Wuhan, that's the large city in China, the 11 million population, using fast, unconstrained Bayesian approximation, as they call it, jokingly, I think, FUBAR, World War II think, analysis is now reported that certain nucleocapsid and spike glycoprotein, those are two of the gene products of the virus, positive selective mutations, I told you it was a positive selection, meaning that it confers some kind of virulent advantage or transmission advantage of the virus, have generated at the protein level, so these are RNAs that turn into protein, they're translated, amino acid substitutions in those two proteins, and they're going to compare that to the previous outbreak coronaviruses. So that's what this paper is about. Good discussion, good understanding of what's going on at the molecular level in terms of changes of this new virus compared to the SARS and MERS. So all the other sites in the N and S genomic regions are highly conserved. That means there's no mutation. Okay, nucleocapsid spike genes, and therefore deemed under tight negative selection. That if you get a mutation there, the virus does not maintain that mutation. That virus with that mutation does not continue on, does not replicate. There are no reported positive selective mutations in envelope or membrane genomic regions. So those two genes, which are also really important for the virus to do its complete systems work inside the host cell, those two don't seem to have any mutations that are specific to the NCIP. Now, this is preliminary data. I'm telling you, this paper's preliminary data, but statistics did seem to be convincing. I looked at them. Even though with a very small n size, that's a huge caveat. It appears they may represent some selective advantage for an epizoonotic transmission, animal to human, from bat to human is what they're thinking here from this paper. Yeah, bat. Bat is not a domestic animal, so the wet market in Wuhan, unless there are bats flying around that wet market, the animals that have the virus could have gotten them from a bat. Okay? So this is where they're leading to. So I want you to understand that this is a different story than what you're hearing on the news. Okay, so the virulent strategy of this transition, substitution of these two amino acids we're going to talk about seem to be related to the effect of NCIP now causing this new outbreak. Okay, and that's comparing the coronaviruses of SARS and MERS. So, indeed, this is a take-home message here. The NCIP, the new coronavirus, looks like it evolved from the bat SARS, it's a unique SARS, like a COV variant, and these new substitutions, amino acid substitutions at the protein level of what? The nucleocapsid and spike proteins, may have decreased disease transmission from bat, decreased disease severity, while increasing disease transmission. But more sequencing of patient isolates have to be performed to verify the supposition. That's what I'm saying. So this is just a preliminary study. Now, within the nucleocapsid, there appears a significant, really small p-value, 0.05. It means highly confident that this is significant. Pervasive episodic selection for two sites, the 380th and the 410th nucleotide positions in the in the Wuhan seafood market pneumonia virus isolate labeled Wuhan HU1. And that's the rest of the designation you can read there. In clade 1, amino acid sequence position number 380, so that's not a nucleotide position, although they are commensurate to that for the DNA level. These are amino acid positions. In clade 1 of this virus, amino acid position number 380 in the Wuhan coronavirus, there is a glutamine substitution for an asparagine. While on the number 410 amino acid residue of that protein, there is a threonine substitution for an alanine. Now, the first one is somewhat conservatory. Second one is certainly not in terms of the chemistry of those amino acids. Now, in another isolate labeled clade 2, the amino acid sequence position number 409, there's a glutamine substitution for an asparagine. Okay. While on the 380, there is a threonine substitution for an alanine. Okay. So you can see there's a slight differentiation there. It's 380, still residue 380, but it's switched in terms of what has occurred there. In the 410 amino acid position, in clade 2, it's a 409 position. All right. Now, within a spike gene product, there appears significant, again, p-value of 0.05, pervasive episodic selection, positive as well, in two sites, 536th and 644th position of the Wuhan seafood market pneumonia virus. The amino acid sequence position at 536, which is related to what nucleotides will code for that. In the Wuhan coronavirus, there's an asparagine for an aspartic acid, while on position 640, there's a threonine for an alanine. Interesting that there's threonine for alanine in a couple of these variants. It seems to be related to the transmission. Okay? Let's move this here. All right. Now, in 2002 and 2003, the SARS coronavirus, severe respiratory illness, affected more than 8,000 individuals with a mortality proportion of around 10%. Not a rate, it's proportion. The subsequent identification of a large pool of coronaviruses circulating in bats and other animals portended the appearance of other highly pathogenic coronaviruses. In 2012, the MERS erupted. That caused a similar outbreak with a higher mortality rate than the SARS. Severe SARS coronavirus infection manifests clinically as acute lung injury associated with high initial virus titer, okay, high viral load, macrophage neutrophil accumulation in the lungs, there's your inflammatory response, and an elevated pro-inflammatory serum cytokines, which are generated from that innate immune cell lineage, interleukins 1, 18, 6, 8, and MCP1 protein. Those are all pro-inflammatory cytokines. Recent advances have implicated inflammatory monocyte macrophages, those are called IMMs, to be class 1 macrophages for you immunologists, in the lungs as critical mediators of SARS-CoV pathology as a delayed type. A delayed type is a different type of interferon responses. Interferon is going to be a key factor here in understanding the molecular pattern of this disease transmission and lack thereof, because it promotes high initial virus titers and an aberrant IMM recruitment. The IMMs, remember, are going to cause inflammation. They are the inflammatory monocyte macrophage lineage of cells that are going to end up in the lung tissues in that bed. Host disease is likely a combination of direct viral damage, viral toxicity, and the consequences, of course, of an aberrant immune response promoted by these inflammatory monocyte macrophage lineages in the lung, underscoring the importance of determining the mechanism of the virus targets innate immunity. Innate immunity seems to be the target here, right? Not acquired. All right. Now, back to a paper published in Cell Host Microbe. Okay, 2016. This goes back now 4 years exactly. Dysregulated type 1 interferon and inflammatory monocyte macrophage responses cause lethal pneumonia in the SARS coronavirus infected mouse model. Respiratory coronaviruses cause acute lethal disease associated with inflammatory responses and lung damage, as we've been saying. A robust viral replication accompanied by a delayed interferon type 1 response signaling orchestrates inflammatory responses and lung immunopathology with a diminished survival in that murine model. Okay? Interferon 1 remains detectable until after the virus titers peak, but early interferon 1 administration in the mouse model ameliorates the immunopathology. So that means that this delayed interferon 1 signaling promotes the accumulation of pathogenic inflammatory monocyte macrophages, resulting in the elevated lung cytokine chemokine levels, vascular leakage, and impaired virus-specific T cell responses, and the impaired acquired immune response. In fact, indeed, genetic ablation of the interferon alpha beta receptor, called IFNAR, or IMM depletion, you can do this in the murine model, protects the mice from the lethal infection without affecting viral load. That tells you the virus replication itself is not causing the disease symptoms, the clinical features. Overall, the murine model demonstrates, from this paper published 4 years ago, that interferon 1 and the IMM, remember those again, are these inflammatory monocyte macrophages, promote the lethal SARS coronavirus infection and identify interferon 1 and the IMMs, as monocyte macrophages, which are inflammatory type 1, as the potential therapeutic targets. Remember, this lecture is about targets in patients infected with the pathogenic coronavirus and perhaps even other respiratory viruses, like for example, the NCIP that we're looking at now. All right, this is the way it goes. If you have early interferon 1 signaling, okay, this is this peak of activity here, and you look at the progression of SARS disease, you get a reduced viral titer, you get a regulated immune response in terms of inflammation, and you get a mild clinical disease. The person cures and survives. Now, if you have the SARS virus building up, then you have a delayed interferon 1 response. You get a higher increase. Look at the arc of the increase in this viral load and the disease that it accompanies it. You get a dysregulated inflammatory macrophage monocyte response. You get severe, because of this IMM, you get lung immunopathology, and you get lethal pneumonia. This is the worst, okay? With no interferon signaling, if the virus doesn't induce it for some reason, or you block it by using drugs in the murine model, you get this huge SARS titer. So it shows you there's the virus, there's plenty of virus. You get no interferon response because you've knocked it out with using, just remember, some murine models, you can eliminate it. And then what happens? Reduced IMM response, no lung immunopathology because you have very low inflammatory macrophage monocytes, and you have the mild clinical disease. So again, no interferon signaling, good in terms of disease for the host. Delayed, the worst. This is when you get death. And early interferon signaling, the whole thing gets blocked. So that shows you some molecular targets. Now, here's a dialectical proposition. This comes from a journal a paper published in the Journal of Neuroinflammation in 2019. There's the rest of the reference. What's the title of this paper? The link between interferon and the inflammasome-mediated damage in various disease states may link coronavirus infection and subsequent disease pathology. See what I'm doing here? I'm going into the scientific literature. I'm giving you the complete aggregate knowledge base on coronaviruses. So we're not just looking at the current one and following those papers, although we are looking at those. What I'm doing for you is evaluating the overall understanding of what coronaviruses are doing inside of cells, what responses are induced in the host, and what molecular targets can be generated, and overall, how you cure without using any vaccine or any drug at all. Why most people cure, right? Because of the immune response. I want you to understand the fundamental basics of immunology If we're going to go target the immune response, we have to understand what's naturally happening in the host when you get a cure versus when you get a mortality or high morbidity. That's what these papers are trying to focus in on, right? Okay, again, this paper was published just last year. So, central nervous system, now just follow along with me what I'm doing. Central nervous system resistant microglia and astrocytes. Now, what are these? These are resident immune cells in the central nervous system, in the brain. Can produce cytokine type 1 interferons. Type 1 interferons, we've already been discussing these for coronaviruses, that signal through the heterodimeric interferon alpha beta receptor. We've already brought this in, the IFNAR. Now, binding of the T1 IFNs, the interferons, with the receptor activates a pathway we've talked about many times in T cell activation. It's the JAK-STAT pathway. That's a phosphorylation cascade, kinase cascade. That leads to transcriptional activation of the interferon-stimulated genes, or ISGs, which mediate both very important paradigmatic consideration here, mediate both pro and anti-inflammatory functions of the type 1 interferons depending upon the cellular environment. And this is why diet event ontological perspectives are so important, and why I put together that paradigm and understanding biomedicine. Depending on the cell type, the same response can give you completely opposing characteristics in terms of, in this case, a particular disease. You understand? So if you're targeting something like interferon, you think, okay, we understand it. We looked at the murine model. We looked at some of the SARS, some of the MERS data. We think we understand replication cycles. Previous discussions on my Authentic Biochemistry podcast and also on BayRef lectures. Then we say, okay, well, then let's make a drug. Let's make something, a biologic or maybe even a vaccine that will then target that specific region of that pathway. And we should be able to get what? Stable decrease in transmission or stable decrease in viral replication or some level of the viral cycle such that we eliminate or decrease the outbreak and overall the potential for an epidemic or even indeed a pandemic. You see, that's how epidemiologists, how governments think. Find a target, go after it, nail it, and then look to see what the sequelae are. But right away, this paper in Journal of Neuroinflammation tells you, depending on the cellular environment, looking at the same pathway, you can have pro and anti-inflammatory responses. You want anti-inflammatory because remember, if you have those IMMs in the lung, that gives you the high morbidity, potential mortality of the coronaviruses, right? Remember that. Okay. Now, indeed, type 1 interferon response in the CNS again, we're in the CNS here with this paper, from viral infections and traumatic brain injuries. You don't need a viral infection to get an interferon response. You can just have a TBI, okay? Playing soccer, for example, right? Heading in soccer. And neurodegeneration can also activate that response. What does that mean? Alzheimer's disease, Parkinson's disease, prefrontal dementia, any number of potential neurodegenerative disease pathways that can occur in the CNS can relate to the induction of interferon. See that? Doesn't have to be a virus. So, type 1 interferons can be protective, of course, or deleterious, not so well known, as in multiple sclerosis, where the type 1 interferons are thought to exert and perform an anti-inflammatory response via the production, here you go, in the MS model, of anti-inflammatory cytokine interleukin-10, with a simultaneous suppression of the pro-inflammatory cytokine interleukin-1 beta. This is why interferon beta is the first-line therapy for multiple sclerosis in the clinical trials, as it limits infiltration of lymphocytes into the brain. First thing it does, because remember these cytokines and chemokines actually function to cause trafficking of immune cells through the BBB, blood-brain barrier. And it decreases the relapse rate. Remember, you have remission relapse in MS. In spite of the evidence that overexpression of interferon alpha gene in the brains of mice associates with neuroinflammation and neurodegenerative disease. Now, see how that looks contradictory, but it's not contradictory because it's different cell lineages. Which ones? Microglia, astrocytes versus what else is in the CNS? The neuron, right? Okay. Obviously, interferon subtype, titration, and cellular residency, neuron versus an immune cell, are critical to this apparent, it's only an apparent or pseudo contradiction. Very important. All right. Now, type 1 interferonopathies are linked to constitutive production of type 1 interferon and an increase in the expression of ISGs. Those are the genes that are turned on by interferon. Type 1 interferonopathies result from mutations. For example, the Aicardi-Goutieres syndrome, which involves the activation of microglia in the CNS and a subsequent chronic neuroinflammation. Other neurodegenerative diseases, the ones just mentioned, AD, PD, MS, and even ataxia telangiectasia, are essentially T1, all four of those, interferonopathies. That's right, Alzheimer's disease, Parkinson's disease, MS. Different way of looking at it, right? Indeed. Because it's now looking at the response to things like a beta protein, tau, microfibrillary tangles, right? Those are the consequence of alterations in protein metabolism. Now we're looking at the other side of it, the interferon response to it. You see, hence an interferonopathy. Neuroinflammation involves activation of an inflammasome, which is itself a cytosolic protein complex that contains multiple copies of what are known as danger sensing receptors. One of them is the AIM2, which we're going to get into a lot of detail in a moment. The procaspase-1, that's kind of canonical apoptosis, and an adapter protein called ASC, which stands for apoptotic speck containing protein. Activation of the inflammasome itself, remember, it involves the NLRP pathway, in glial and neuronal cells results in caspase-1 activation, pyroptic cell death, that's a specific fever-associated cell death. It's not like regular apoptosis in terms of its cellular morphology and clinical sequelae. Also, this whole pathway results in the release of pro-inflammatory cytokines like the really potent IL-1 beta and interleukin-18. All right. Now, I want to give you a distinction of terms here. This is what I do in authentic biochemistry. Right. This comes from a paper way back, Nature Review Microbiology 2009, so 11 years ago, February 7th, 11 years ago. It's very important to go back, do a deep dive into the primary refereed scientific research, that component of the biomedical literature, to be able to get a complete understanding, an architectonic, if you will, of these diseases, even something brand new and incipient like NCIP. So here's a paper 11 years ago. Apoptosis, programmed cell death, sensu stricto, requires a particular cysteine-dependent aspartate-specific protease. Those are called caspases. Okay? They produce an orchestrated disassembly of the cell as they cleave cellular substrates, resulting in cytoplasmic and nuclear condensation, DNA cleavage, make a specific ladder conformation, and maintenance of an intact, this is really important, plasma membrane. The degradation products of apoptotic cells are packaged into membrane-enclosed bodies that are targeted ultimately for phagocytosis and removal in vivo. That results in the absence of inflammation because you keep everything closed up, right? You do not release anything that's going to induce an inflammatory response, proteins, lipids, carbohydrates. Uniquely, caspase-1, a specific caspase, it's still a caspase, canonical for apoptosis. Caspase-1 dependent programmed cell death has a different subtitle, subtext, and it's called pyroptosis. Pyroptosis. Programmed cell death is stimulated by a range of microbial infections. For example, Salmonella, Frangiella, Legionella, and non-infectious stimuli, including host factors produced during an MI. What's that? Yeah, myocardial infarction, a heart attack, wherein caspase-1 processes the inactive precursor of interleukin-1 beta and interleukin-18 into the mature inflammatory cytokine. So here's caspase-1 taking a preformed pre-pro inflammatory cytokine and converting it to a pro-inflammatory cytokine, giving you the full-blown inflammation. You see? That's what that caspase-1 is doing here. And what subsequent to that? Rapid cell death characterized by plasma membrane rupture, release of the pro-inflammatory intracellular content. So pyroptosis is nothing like apoptosis. Apoptosis conceals all of this self-destruction, eliminates the virus or eliminates the damaged cell, and then goes on to have that phagocytosis and elimination of any inflammatory response because it's never triggered. Pyroptosis is totally different. The cell starts off with a programmed cell death, but because caspase-1 is mediating it, you get the production of these pro-inflammatory cytokines from the pro-inflammatory cytokines, interleukin-1 beta and interleukin-18. And that results in a complete annihilation of the cell, the opening up of the plasma membrane, the secretion and loss of those pro-inflammatory cytokines, and you're on your way then to a tremendous rapid cell death that is now spreading. If it's in the CNS, of course, it's quite dangerous. Membrane rupture, which is previous to that, of course, and then release of those pro-inflammatory cellular contents. It's not just the cytokines, but proteins themselves can act as antigens. Now, I want to introduce a new gene. It's called AIM. Okay? So this slide here I'm calling Take AIM. This comes directly from gene cards. So you can go online, you can look up genes, and you can look at what the genes have been described as in the scientific literature. So it's a conglomeration of a bunch of information of any gene that has ever been recognized as specifically or often associated with biomedicine. So AIM is one of them. What's AIM? AIM is apoptosis inhibitor of macrophage. Right away, inhibitor of macrophage. It's key. It is a secreted protein that acts as a key regulator of lipid synthesis. That's what I said. Remember, I'm a lipid biochemist. Mainly expressed by the macrophages. These are innate immune cells, right? In lymphoid and inflamed tissues. And it regulates mechanisms of inflammatory response, such as infection or even atherosclerosis. So AIM has a tremendous literature to it. AIM decreases, now here we get back into the lipid metabolism, lipid droplet size in adipocytes. That means the triacylglycerol lipid droplet size, its diameter is diminished because of AIM. Following incorporation into mature adipocytes via the CD36 mediated endocytosis of fatty acid, associates with a cytosolic FASN, which is an inhibitory fatty acid synthase activity. FAS stands for fatty acid synthase. FASN is something that inhibits fatty acid synthase in the cells. From AIM, that leads to lipolysis. So rather than lipid synthesis, lipolysis, break down the triacylglycerol. And, as I just say there, the degradation of triacylglycerol into glycerol-free fatty acid. That's what happens because of AIM. You don't get lipid anabolism, you get lipid catabolism, leading ultimately, presumably, to beta oxidation of fatty acids for the ATP synthesis pool. Now, AIM, which is actually from a gene called CD5L in humans, induced lipolysis occurs with progression of obesity, participates in obesity-associated inflammation, or OAI, following recruitment of inflammatory macrophages, remember the IMMs from the coronavirus discussion, directly now into adipose tissue, not into the CNS, not into the lung, but into the adipose, visceral fat, for example. That causes an insulin resistance because you're disrupting all the receptors in the adipocyte, and obesity-related metabolic disease like metabolic syndrome or type 2 diabetes, where the regulation of intracellular lipids mediated by the CD5L, which is the AIM gene. It has a direct effect of transcriptional regulation mediated by nuclear receptors such as the what? Retinoic acid orphan receptor gamma, or the RORC. Talked about this before. It's a transcriptional factor, right? Transcription factor generates different gene expression products because you turned on the ROR gamma. Okay. Now, that's what AIM does. AIM functions as a regulatory of metabolic switch in Th17 cells. We've talked about these many times in Authentic Biochemistry and VeraMed, where it regulates the expression of pro-inflammatory genes in Th17 lineages by altering the lipid, yes, I said lipid content, and limiting the synthesis of cholesterol ligand of RORC. Okay. And that's the master transcription factor of Th17 cell differentiation. After all, cholesterol is mediating Th17 differentiation. Did you know that? Did you know that cholesterol mediates T cell differentiation? If you didn't, now you do. AIM, so cholesterol is not acting as a membrane factor, just as a membrane component altering membrane fluidity in the plasma membrane. It's certainly not acting as a precursor to steroidogenesis, for example, or other prerenal lipids. Here, cholesterol is mediating transcriptional alteration in what? In Th17 lymphocytes. So AIM is mainly present in non-pathogenic Th17 cells where it decreases the content of polyunsaturated fatty acids affecting two metabolic proteins, the MSMO1 and the CYP51A1. That's a cytochrome mediated response. And that synthesizes ligands of RORC. That's how this whole process is linked together. Okay? Thus limiting RORC activity and expression of the pro-inflammatory gene. So that's actually how it works. Mediates through decreasing the content of polyunsaturated fatty acids, which then decrease the activity of two metabolic proteins shown there. Then, because those normally synthesize ligands for the RORC, because those ligands are not being now synthesized because of this diminishment of polyunsaturated fatty acids, presumably because of beta oxidation of fatty acids, presumably because AIM is carrying out catabolic events in lipid metabolism, you get a decrease in the expression of pro-inflammatory genes in Th17. This Th17 is standing down. You see? AIM also participates in obesity-associated autoimmunity, big topic in Authentic Biochemistry and VeraMed recently, via its association with IgM, it's a specific class of immunoglobulin, membrane-associated, of course, interfering with the binding of the IgM to the Fc alpha mu receptor and enhancing the development of long-lived plasma cells that produce high-affinity IgG autoantibodies. Wow. So it's even involved in plasma cell autoantibody production. So you can see here that AIM has multiple roles in pathology, okay? Particularly in clinical manifestation of pathology in humans. Now, finally, one more thing. And finally is a word I would put in quotation marks because we're not certainly done with AIM. Finally, AIM acts as an inhibitor of apoptosis. Yep. In macrophages. Now, we're not in Th17 cells, we're in macrophages, back to innate cells, because it promotes macrophage survival from the apoptotic effects of oxidized lipids in case of atherosclerosis. And further, AIM is involved in early response to microbial infection against various pathogens by acting, oh my gosh, as a pattern recognition receptor because it promotes autophagy rather than apoptosis. Okay? That's a key feature here. So, take a look at these slides. Traumatic brain injury, pyroptosis, or an infection can lead to an alteration in DNA being released from the nucleus. That then will interact with microglia. Now, look what happens. This is a murine model. The murine microglia express detectable basal levels of AIM2. Basal levels. Detection of low levels of cytosolic DNA released in the brain due to TBI, pyroptosis, or an infection. Okay. And of course, this pyroptosis involves the inflammasome, an LRP pathway. And any other kind of brain infections, like a coronavirus or other kind of virus, activate the interferon stimulatory DNA, or ISD pathway. That's over here. Okay. The T1 interferon response results in the upregulation of the AIM gene, an anti-inflammatory cytokine, cytokine IL-10, which is right here, is generated. Okay, that's normally what happens with low DNA. Now, detection of an increased level of DNA, because increased traumatic brain injury, increased pyroptosis, increased infection, bacterial, viral, increases the amount of cytosolic DNA because you're getting nuclear damage by them, or sometimes because of replication patterns of certain viruses or even bacteria. By the macrophage microglia activates the AIM2 inflammasome, resulting in the production of the pro-inflammatory cytokines we just talked about. So now you're interrupting the AIM2 pathway. Now you've got an AIM2 pathway inflammasome. So you've flipped its activity from being anti-inflammatory to pro-inflammatory in the same cell lineage. Now you get, because of that caspase-1 mediated response, the production of pro-inflammatory interleukin-1 beta, interleukin 18, that leads to an AD-related inflammatory response, such as Alzheimer's disease, neurotoxicity, neurodegeneration. And the only thing that was going to block that residently in the CNS is going to be interleukin 10, which is going to inhibit all that, and the Treg cells, which is another whole topic. So the activation of the AIM2 inflammasome and pyroptosis is going to lead to more pyroptosis, more DNA being built up, trying to now knock down this whole pathway by increasing the amount of interleukin 10 that's inhibiting the AD, the Alzheimer's disease-related inflammation, neurotoxicity, neurodegeneration. So you've got two things going on in one. You've got a partial amelioration because you've got the ISD pathway making T1 interferons, or you've got the full-blown AIM1, AIM2 inflammasome flipping the AIM response, production of the caspase-1, AD-related inflammation and onwards. Interleukin 10 still gets generated from this back pathway because it bifurcates, it gets away from the inflammasome. In fact, the inflammasome produces more pyroptosis, which makes more DNA, which can then go not through this pathway, but directly to the ISD pathway, making IL-10 blocking it. Okay. Cell death by pyroptosis inhibits the ISD response pathway and the production of IL-10. So the more cell death you get, the more this shuts down. The more this shuts down, the higher the inflammation rate. That's what everything's about. Now, AIM2 and AD, Alzheimer's disease-related inflammation and neurodegeneration. Once again, look at this. Traumatic brain injury, pyroptosis, or an infection. You get an innate immune response, microglia, astrocyte. You get now type 1 interferons. They're going to produce AIM2. They're going to produce another protein called P2O2, which is also something that's generated naturally, depending on the mouse strain, whether that's male or female, and what cell type. All of that's going to block AIM2. So interferons of the type 1 lineage will produce 2O2, as will the normal genetic machinery, which will block an AIM2 activity. When you block AIM2 activity, you block this pyroptosis. So you see AIM2 in the presence of DNA will generate that AIM2 inflammasome, make those pro-inflammatory cytokines, cause pyroptosis, and you're off to having full-blown inflammation. At the same time that's happening, type 1 interferons are making P2O2, which is blocking that pathway. That's the important feature here I want you to understand. This is where AIM2 can be related to inflammation and neurodegeneration, depending on how much of it is made. Okay? So the higher P2O2 to AIM2 protein ratio in microglia, the CNS of Alzheimer's patients, is predicted to suppress Alzheimer's disease-related inflammation and maintain homeostasis in CNS. So it's two-way. Now, finally, knockdown of the AIM2 protein. In human normal lung, now we're in the lung, fibroblast, or a deficiency of the AIM2 gene in the murine bone marrow derived macrophages, which where they come from, activated ATM protein kinase, that's a member of the P13 DNA protein kinase family, which can bind to AIM2 protein, thus suggesting a role for the AIM2 protein in the inhibition of the ATM protein kinase activity. A dysfunction in the ATM, okay, down here, ATM, protein kinase and microglia accumulated DNA did not result in activation of inflammasome or indeed pyroptosis, okay, because you're blocking it with AIM2 here. So again, you've got the innate immune response making type 1 interferon, Type 1 interferons making AIM2. That can trigger an AIM2 inflammasome, pro-inflammatory cytokines, pyroptosis. Same time, AIM2 can do that directly without going through the inflammasome. But it also blocks this ATM, which would normally inhibit, right? And therefore induce this CGAS STING TBK pathway, which would then trigger more type 1 interferon production. So the type 1 interferon response, which upregulates the expression of the AIM2, shown here, is predicted to prime microglia for activation of the AIM2, the ATM system, inflammasome, upon sensing, it's up here, upon sensing higher levels of cytosolic DNA. You always need a lot of cytosolic DNA due to, again, traumatic brain injury, pyroptosis, or infection that results in an Alzheimer's disease-related neuroinflammation, neurotoxicity, and ultimately to neurodegeneration. So these are other genes involved. The CGAS STING TBK pathway, further triggering these responses. Okay, and you're blocking this ATM inhibition, which would erstwhile shut all this down. So let's make a synopsis here, important at this point of a lecture. Sorry. Suppression of the type 1 interferon by AIM2 and AIM2 proteins in the CNS may be important to keep the levels of type 1 interferon-inducible negative regulator proteins like the murine P2O2, and humans have an analog to this, the IFI16 beta and the POP3 gene products, below a threshold to suppress neuroinflammation related neurodegenerative diseases. So suppressing the type 1 interferon response by AIM2 can give you an amelioration of Alzheimer's disease. That's what this is saying, because of that feedback mechanism. The generation of the AIM2 deficient Alzheimer's disease mice on the pure B6 genetic background, this is a specific background looking at Alzheimer's disease in the murine model, provides a novel insight into the precise role of AIM2 proteins in the modulation of neuroinflammation. Remember, it can go one way or the other in the development of Alzheimer's disease. Now, an improved understanding is what this paper and this whole lineage of papers is talking about. Again, more research is needed. Of the mechanisms of which absent in melanoma, that's AIM2 proteins, okay, and their negative regulators modulate neuroinflammation, could identify new approaches to treat AD. Now, why am I putting all this together? So that you understand that these proteins have multiple roles. Remember, I said take AIM. We started to get into AIM. I took deeper cut into AIM to show you this is a major molecular target to understand Alzheimer's disease because it's a neuroinflammatory response. Inflammatory response. I'm just showing you here that depending on the timing, tissue type, and alteration of transcription factors as related to fatty acid metabolism, lipolysis versus anabolic triacylglycerol synthesis, CD36 mediated fatty acid introduction to those cell lineages, which is linked to obesity because of the insulin insensitivity, thus increasing the utilization of fatty acids, high circulating fatty acids associated with that obesity because of high circulating what? Lipoproteins loaded with lipid, right? Particularly with fatty acids. And CD36 orphan receptor taking those fatty acids in, making more triacylglycerol, exacerbating the problem, for example, with insulin resistance. All of that can be related to Alzheimer's disease and also related to viral infection through the mediation of these molecular targets like AIM. There you go. Now, we'll stop here because I want to get on to another whole chapter of a discussion of how I'm going to synthesize this information and get back to coronaviruses. Okay, we're going to look at a paper published in Nature Review of Gastroenterology and Hepatology. You can see here we're going to start talking about primary biliary cholangitis. Okay, and ultimately we're going to end that whole system by going back to this coronavirus, back to interferons. You see, that's what I'm going to be doing next time. So I'm going to stop here now. This is my stop page because I didn't want to continue on with that whole discussion because I didn't want to keep you from what you're normally doing on a Sunday afternoon. This is a Sunday afternoon. But I want to make sure that I got this initial phase of this lecture out. So again, I'm Dr. Dan Guerra. This is an authentic biochemistry video lecture. I promise you we start to do that. And this is just authentic biochemistry. This is February 9th, 2020. And there's my Facebook page where you can find, this is where this is going to appear via the feed that comes through Facebook that ultimately comes through the video lineage. You can also find me again on by subscribing to that YouTube channel, which is where these things are ultimately posted up, and the Apple Podcast, Authentic Biochemistry, where I will then incorporate what we're talking about here into podcasts. Now, what I don't do in my Authentic Biochemistry, I don't tape this, excuse me, and then just play the audio for Authentic Biochemistry Podcast. No. All my Authentic Biochemistry, both the video you just watched and all my Authentic Biochemistry audio feed are unique. So whenever you hear Authentic Biochemistry Podcast, it's going to be reflective of this video lecture, incorporate what we talked about, and moving further on. I'm going to go back to a video next time for Authentic Biochemistry, because you saw those slides I just ran through, that little lineage. I want to take you carefully through the rest of that pathway to give you the complete diet event ontological perspective of how these various molecular targets are associated with the NCIP coronavirus as potential ways of understanding first the immune response, the low possibility of having high mortality with this new virus, because it seems to have very low virulence related to high mortality, but perhaps an enhanced transmission because of those amino acid substitutions we talked about in the nucleocapsid and in the spike protein, right, which were positively regulated in terms of evolution of that virus. All right, so I think we got there. Now what I want to do, like I do every time when I finish my talk, is I want to say thank you very much for listening to my Authentic Biochemistry lecture. I'm going to come back with another video lecture to cap off this region of this discussion, and then we'll move over to having podcasts, just the audio, for those of you who just listen to audio, summarizing once again very briefly what we talked about in this long-term video lecture, asking you to go back and look at these, and then ultimately what? To synthesize a good understanding of the molecular targets that may be related to this coronavirus outbreak, and even a better understanding of diseases associated with inflammation, autoimmune diseases, neuroinflammatory diseases, lung diseases with or without the inculcation virus, like in the coronavirus. So what I want to say finally, with that long ending, is have a really great Sunday, the 9th of February, 2020. This is Dr. Daniel Guerra saying bye.